Hi engineers, in this video today we're going to be talking about the SARS-CoV-2 variants. Before we get into this video though, please continue to support us by hitting that like button, commenting down in the comment section, and please subscribe. Also, down in the description box, we'll have links to our Facebook, Instagram, Patreon account. Go check those things out. All right, engineers, let's get into it. All right, engineers, so in order for us to understand these SARS-CoV-2 variants, we first have to take some time to talk about the original D614G SARS-CoV-2 virus and talk about its kind of replication, life cycle. And then once we do that, then we'll talk about the variants and what are the genetic changes or mutations that we're seeing that kind of contrast from the original SARS-CoV-2 virus and how those mutations may make the virus a little bit more fit in the sense that it increased transmissibility, increased viral production, increased viral replication. And the question is there increased lethality, severity, mortality associated with these variants as well? So let's have to kind of take a quick kind of stroll through the replication or life cycle of the original SARS-CoV-2 virus. So there was a primary protein that has become the most kind of concerning protein on the virus. What was the name of that protein? It's this red protein here. Do you guys remember? It's called the S protein. We commonly refer to it as the spike protein or S protein. This is primarily the one that's of concern. It's the one that whenever they were making vaccines, it's the one that we're primarily developing an immune response against, okay? That spike protein is expressed on the outer part of the virus. But you have to remember again that this virus is an envelope. So this blue structure there just represents the envelope, which is kind of like a phospholipid bilayer. Then what's important is inside of that virus you have a bunch of proteins. This purple protein is called the nucleocapsid. Okay, so again, you have your spike protein, and then you have this blue structure here, which is called the envelope. And then you have this purple structure here, which is kind of your nucleocapsid. And then inside of that nucleocapsid is going to be the genetic material. And in this case, this is the RNA. But particularly we call this single-stranded RNA. And it's a positive sense single-stranded RNA that we see inside of this SARS-CoV-2 virus, the original one. The one that was primarily responsible for the global pandemic. What we know from previous videos is that whenever the spike protein, in order for it to actually help the virus to get into the cell, it has to bind to a particular protein that's expressed on cells in our body. And the primary one that we saw a lot related to this was the respiratory tissue, particularly like the alveoli. Remember within the alveoli you had the type 1 alveolar cells, the type 2 alveolar cells. We were seeing a lot of actual infiltration of that virus into those tissues. How did it get into those cells though is the question. And if you remember, the spike protein had to bind to this pink protein here on the cell surface called an ACE2 protein. And that ACE2 receptor, ACE2 protein, would bind to the spike protein and activate this process. But then the next thing had to happen. You have this green protein on the side of the ACE2, and that's called TMPRSS2. It's a transmembrane protease. And what this does is it cleaves a particular part of the actual viral proteins that helps to engage and bring the virus into the cell. So again, once the virus has its S protein bind with the ACE2 and use this transmembrane protease to cleave a particular site on the viral peptide, it then causes a receptor-mediated endocytosis, which brings the virus into the cell inside of like a little vesicle, okay? Once inside of the cell, you'll have to release that RNA, that single-stranded RNA, out of that vesicle and into the cytoplasm where it can then interact with our ribosomes. What we need to do is take a look at this positive sense, single-stranded RNA, and kind of take a look at this genome a little bit. There's a lot of different genes that you can see on this RNA strand that we're going to translate with the ribosomes, and they make proteins. And a lot of these, like this blue one here, this may give rise to particular types of molecules, such as like polymerases. Like you have RNA polymerases, and you have replicases. These may be proteins that are actually going to be coming from this particular gene, from a particular open reading frame. Then from another reading frame, this may give rise to other types of molecules called proteases. And the whole significance of these is that these RNA polymerases and replicases are important because they help to take the RNA here and make more RNA so that we can make more virus. The proteases break down specific polyproteins that also help within making particular activated viral proteins. Then you have this other one here. We're going to save this black one here because it's the most ominous. This one here on the side, this brown one, may give way to particular proteins that are expressed on the virus that we're not super concerned with. We've already talked about a lot of these. We're going to abbreviate them, but they're like the envelope proteins, okay? The nucleocapsid proteins. And there's even other ones. These little brown ones there could be what's called hemagglutinin esterase proteins. These are proteins that, again, can be kind of exposed on the virus as well. But these aren't the ones that we're super concerned with. The one that we're most concerned with respect to the SARS-CoV-2 virus is this gene that codes for the spike protein. That codes for the spike protein. And the reason why I'm kind of taking the time to mention all this is that the SARS-CoV-2 variants are experiencing mutations in this particular gene. And these mutations alter the structure of the spike protein that in some way it may help to increase the viral entry into the cell or particularly evade the immune system. And that's why it's a concern. And we'll talk about how and what those mutations are. But again, the whole goal here is that these genes will express RNA polymerases so that we'll take some of this single-stranded RNA and we'll replicate it and we'll create more single-stranded RNA. So now I have a bunch of single-stranded RNA. Then I'm going to take these proteases, which are going to break down large amounts of polyproteins, which are going to also give way to particular viral structures. Then I'm going to have my spike protein. And what was the color of my spike protein? Just to be consistent with the diagram here. This was that red protein there. That was the main one. And then we're also going to give way to some of these other proteins. And what were these? These were like your envelope protein. And we're just going to represent this like blue in this case. Maybe you have a blue envelope protein. Then you have an N protein, which is the nucleocapsid. We'll just represent that as purple. And then you even have the hemagglutinin protein, and again, that could be a different color as well. But again, the whole point here is what I want you to understand is that from this SARS-CoV-2 genome, we made spike proteins and we made more single-stranded RNA. These are the two dangerous things that we're worried about. What happens here is that from this, we're going to take the RNA and all the viral proteins and then ship this to what structure? We're going to take all of this stuff here and ship it to the Golgi apparatus. Once we send it to the Golgi is going to start packaging all of these molecules into a vesicle. And out of this vesicle, you're going to start forming parts of your virus. And so we'll kind of quickly put together here, let's say that here's our envelope. Okay, here's our nucleocapsid here. If we were to draw another one there. And then here we're going to have the primary protein that we are really concerned with here is these S proteins coming off. Okay? And then again, what's inside of these viruses, if you're making tons of them, you're making a lot of this single-stranded RNA. And then again, this would all be kind of packaged into a vesicle by the Golgi. And then what would happen is this membrane, this Golgi membrane would fuse with the cell membrane and then release this virus out of the cell and then go and infect other cells. In the process, it's going to cause virus particles to build up in this cell and eventually destroy this cell. So this covers our basic life cycle or replication cycle of the original kind of SARS-CoV-2 virus, primarily putting an emphasis on what? The particular portion of the SARS-CoV-2 genome is the part that expresses for the spike protein. We're seeing particular concerns and mutations that are occurring right here in that spike protein. Changing the spike protein morphology could potentially lead to evasion of the immune system and increase infection, replication, production, and spread of the virus. How? That's the next question we have to address. All right, so we understand now the general, very basic understanding of the replication of the SARS-CoV-2 virus, right? Again, the original one. What we now have to understand is how is these variants, particularly seeing mutations within the spike protein causing a change in the life cycle, the replication cycle, the infectivity, the transmissibility, and how is that different from the original SARS-CoV-2 virus? Okay? So first thing we have to understand is what these variants are, and we're giving specific names to them. The general name for this first variant here, B117, is actually going to be the one that originated kind of in the UK, so we kind of call it the UK variant. And it's starting to become one of the more prominent viral variants and may be becoming the most dominant one globally in the future. The next one is B1351. This is the South African variant. And we're seeing some spread with this one, but again, originated kind of in the South African region, and we're seeing some spread globally. And the last one here that is a variant of concern is P1. And this P1 is the one that kind of originated in Brazil. And again, we're not seeing a significant amount of spread of this one, but again, still potential that it could spread globally. But again, these are the variants of concern, or VOCs as we say it. What we need to understand is what makes these variants a concern. And that comes down to these particular mutations. And that's what I want to kind of spend a little bit of time on, talking about the science, the genetics, the immunology and stuff behind it. So the first thing that we have to know is, again, this is all relevant. Let's imagine that if we were to kind of zoom in on just that gene. So again, we're zooming in on this particular genome here. And this is just the spike protein gene. Just this entire length here, from this portion here to this portion here, this is the spike protein gene. Okay, and it could be longer. We're only really focusing on a couple particular portions that we're seeing mutations in within that spike protein gene. And it's a pretty long genetic sequence. The first one that we see a mutation in is N501Y. This is the particular mutation we're seeing. It's basically a substitution between amino acids. Within the 501th amino acid of the spike protein, we're seeing a substitution. Now, that's not the important part. The important part is, what is this mutation leading to? If we look here, this first one here, the first portion where there's a mutation in the spike protein gene, the N501Y. When there is this mutation, it leads to a substitution, and this substitution leads to a change in a particular part of the spike protein. Now, something that we haven't talked about in our videos for the SARS-CoV-2 virus is the spike protein structure. It actually has three different components, if you will. One component is the S1 component, here in the dark red. Then the one in white is the S2 component of the spike protein. And this last little kind of ball on the top is called the receptor binding domain. These are the three portions of the spike protein. Now, what we're seeing is, in the spike protein gene, if there is this particular mutation, it changes the structure. This kind of substitution in amino acids leads to a change in the structure of the receptor binding domain. And just a little change. But what does that change in the receptor binding domain do? Let me explain. Let's imagine that we just zoomed in on that spike protein particularly, and it's going to bind here. So here's the two portions here. We're going to kind of have this portion here, this portion here. The receptor binding domain is actually the particular part that interacts with the ACE2 receptor. So the receptor binding domain interacts with this particular portion of the ACE2 receptor. This mutation in the original SARS-CoV-2 variant, particularly to the UK variant, increases the entry, binding, and kind of endocytosis of the virus into the cell. So that receptor binding domain, the mutation, increases the affinity or interaction between the S protein and the ACE2 protein, and increases the entry of the virus into the cell. What would that do? If you increase viral entry, what would that do? Well, now you have more virus coming into the cell. If you have more virus coming into the cell, you can technically increase the viral replication then. And if you increase the viral replication, what could that potentially do? You could increase the viral infection within the particular area of wherever this infection is occurring. In this case, the lungs. Then if you're particularly infecting that cell with lots of viruses and you're replicating and making more virus, that can increase the viral production. So now not only are you taking in more viruses, you're replicating more viruses, you're infecting more cells, and you now have the ability to produce more virus. That could be a potential problem. Why? If you have more viral entry, more viral production, more viral replication, what that might mean is that you have more viral load within you. And that higher viral load, it would be easier for you to just kind of sneeze, cough, cough on some surface or directly onto a person and then hit one of those areas of the mucous membrane. A higher viral load means that you might inoculate the individual with more virus. That could potentially increase the transmissibility of the virus. So that may be one particular concern, is that you can therefore increase the transmission of the virus. Beautiful. But the whole primary thing to focus on here is that the concern is that this mutation in the receptor binding domain will increase viral entry, more virus coming in, more replication, more potential infection of the tissues it's invading. You can produce more, infect more cells, and then spread more potentially. All right, the next mutation that we see within B117, or the UK variant, is particularly a deletion. You're deleting an amino acid 69 and 70 within the S protein. And now, if we were to kind of highlight that, particularly here, this red portion here, there's a deletion, HV6970. And this would be like, if you really wanted to know, these are amino acids. You just have to look at their single letter abbreviation. Like, for example, H is like histidine and V is like valine. So there's a deletion in those amino acids. Now, again, let's say here's the S1 protein, here's the S2 protein, here's the receptor binding domain. That deletion changes a particular structure within the S1 or S2 peptide. It changes just a little bit of it. And that could be potentially a problem. Why? You know, whenever antibodies are produced, whenever you generate an immune response, so let's say that your cells get infected, generally your immune system will get activated. And eventually your plasma cells will produce antibodies. Those antibodies are going to be generated against the S protein. Well, the antibodies that you generate, let's just say, for example, you've already received the vaccine, the Moderna, the Pfizer vaccine. You've produced neutralizing antibodies to the S protein for the original SARS-CoV-2 variant, or you've been infected with SARS-CoV-2, the original one, and you now have a natural production of antibodies. The concern is that these antibodies that you've produced, whether it came from the vaccine or whether from natural immunity, that this portion of the antibody that binds with this S protein, because of that small little mutation, that that antibody may not be able to bind to that portion of the virus, viral S protein specifically. That may lead to, the worry is evading your immune system, evading the neutralization of antibodies. So what is the concern with this? The concern with this deletion is that this may lead to evasion of neutralizing antibodies. And here's the thing though, let's say that you do evade. Let's say that in this person who has been vaccinated, so again, this would be vaccinated or natural kind of immunity, and you generated these antibodies that are going to be directed against this S protein, and it's a small change from the original. One other quick thing, these antibodies, you know how many amino acids it takes for these to interact? It takes about 6 amino acids. So in order for an antibody to bind to this particular antigen, it needs about 6 amino acids to do that. Here's 2 things I want you to understand. We only made a 2 amino acid change. That's the first thing. It takes about 6 amino acids for that actual antibody to bind with. So this interaction may not be as strong, but there still could be some interaction with these antibodies that you generate from vaccination or natural immunity. The second thing is, this S protein is a big son of a gun. So even if you aren't even if there is a mutation in this small portion here, you still have many other areas that the antibody can bind with. So that's the important thing. But let's say worst case scenario, your neutralizing antibodies somehow don't work on this S protein. Guess what we have though, and this is why I think it's very important to understand, your immunology. We still have our memory T cells and our memory B cells. I don't think that these variants are going to be able to bypass that system. So because of that, even if there is an invasion of the neutralizing antibodies, and we'll talk about this a little bit later, the question is our memory cells, which ones? T cells and B cells, are they enough? And I truly think that they are. So that's the next thing, that's the next concern, okay? All right, one more mutation. And you know what's beautiful about this? Is that if you look here at the B117, look at the N5019 mutation. It's present in the South African variant and the Brazil variant. So all of these potentially have this, this mutation, this change that we're actually concerned with. Okay, so this next one that we have to talk about that we really only see in the UK variant is this substitution at the 618th amino acid of the S protein. And there's a substitution between P and H. So, and again, you have to look at the single amino acid abbreviations, but again, it's P618H. So what is the worry about this particular mutation? The worry is that again, it changes a particular structure in the S1 or S2 peptide portion. So here's S1, here's S2, here's the receptor binding domain. The area that we may see a change is in the S1, S2 portion. Why is that a concern? Let me explain. So let's say again, we only focus on that actual portion here binding. So we have the receptor binding domain that's binding here, and then we have the two portions here, the S1 and the S2. Remember, the receptor binding domain is the part that we really want to focus on with the ACE2 interaction. But remember this protein here, it was green over there, but we're just calling her an orange for keeping this consistent with the mutation. It was that TMPRSS2, that transmembrane protease. Do you remember what it did? It led to cleavage of a particular part of like the S protein that helped to increase the receptor-mediated endocytosis, taking the virus into the cell. Well, these mutations from this P618H mutation, it may increase the activity of this cleavage site. If you increase the activity and interaction with this enzyme and you have this increased cleavage, what is this going to do? It's going to bring more virus into the cell. So the concern here with this mutation is that this may increase the cleavage activity, so it increases what's called furin cleavage sites. And the concern with that is that it's going to increase viral entry into the cell. I want to talk about this last mutation that we only see in the South African and the Brazil variant. So, okay, real quick recap. With the B117 or the UK variant, the primary mutations are in these three. And what was the overall consensus? This one increases the receptor binding domain ACE2 interaction, increased viral entry. The deletion potentially changes a spot on the S1, S2 peptide and potentially may evade neutralizing antibodies. The P618H increases the activity of the furin cleavage with the TMPRSS2 protein. That increases viral entry into the cell. The South African and the Brazil variant, guess what? They both have that N501Y1. So again, it's the same process, increasing the viral entry. What do they have though that's different in comparison to the UK one, this pink one? The E484K mutation. And again, it's a substitution here. And again, if we look at it here on this particular portion of the gene, it's this E484K. So at the 484th amino acid, you're having a substitution. Now, where is this substitution and why is it a concern? We've kind of already hit this, so it's going to be a pretty quick recap. Again, what is the structure here? S1, S2, receptor binding domain. Now, if I'm a little bit particular, just a teensy bit more specific, The receptor binding domain has a particular portion of it that really is kind of coming in contact with the ACE2. It's the most intimate contact point between the receptor binding domain and the ACE2. And this is, it's called the receptor binding motif, if you really want to be specific. What we're seeing with this mutation is that there is a particular change in the structure of the receptor binding domain, but particularly within the receptor binding motif that's a part of the receptor binding domain. What the heck does that do? Well, let's talk about it. All it does is, let's actually color code it here. What do we have here? This is blue, we said. This right here, you're going to have very intimate interaction now between the receptor binding motif of the receptor binding domain and increase interaction with the ACE2. What will that do? Increase viral entry into the cell. If you increase the viral entry into the cell, what is the problem and the fear with that? We already talked about it. If you have more virus coming into the cell, it's a simple process here. Think about it. More virus coming into the cell, okay? That virus is going to replicate, make more RNA, make more proteins, S proteins in this case. So let's actually color coordinate it. You're gonna make more S proteins. If you make more S proteins, more RNA, and you combine both of these together, what are you gonna do? You're gonna make more viruses. We'll just represent it like this, okay? If you make more and more and more viruses, guess what that's gonna do? That's gonna go and infect more cells, cause damage to more tissue cells, and increase the viral load within the individual, which can do what? Potentially increase the transmission of the virus. So this is why there is this fear of these viral variants. Now, I'm not gonna lie to you, there is certain research out there that says that the transmission of the virus with these variants is increased, okay? So maybe up to potentially 30 to 50%. So that is a lot. But here's the thing I don't think we have enough research on. And that's the question I think we need to kind of have a little bit of understanding is okay, yes, you can have increased transmission of that virus, these variants. But what if it's asymptomatic? What if it's mild cases? You're not getting a lot of those moderate to severe to fatal cases. That's something I don't think we know yet. I do think that if you think about this, the more viral infection you have within a population, there is just going to be a higher incidence potentially of fatality. But within a general population, I don't know how much more fatal these can be. I still think that if you have been vaccinated or that if you've already been affected with the original SARS-CoV-2, I still think you're going to have enough memory T cells, B cells, and for the most part, neutralizing antibodies. The only thing I will say, and I'll explain it a little bit later, that I think could be debated is if you were infected and it was a very asymptomatic or mild case. Maybe you didn't generate a powerful enough memory cell response or a powerful enough antibody response. If you were infected with a high viral load from one of these variants, there could be the possibility that your immune system won't be strong enough in that sense, if you think about it, to fight that variant off. But again, I still think that we have protection from these variants if you're being vaccinated, and if you have the infection, you also should have some general immunity. And again, we'll talk about some other ways that you can optimize your immune system. But this, I think, kind of gives us the genetics behind the viral variants. Now what I want us to do is talk about the immunology. Is vaccines and is the natural kind of immunity that we developed if we've been infected with the original SARS-CoV-2 variant enough to battle off these variants? The question is, and this is something that I would want to know, and I hope that it's something that you guys were thinking about, is again, if we've been vaccinated with what? Well, the big ones that are out there right now is the Moderna and the Pfizer. And again, there's also other ones out there, like the AstraZeneca Oxford vaccine, the Johnson & Johnson's coming out, the Novavax. There's a lot of different vaccines, but these are primarily the ones that most people have received with respect to the original SARS-CoV-2 virus, before these variants really started popping up. The question is if you were vaccinated, would you still be able to get infected with these variants? And here's why I would say for the most part, no. Okay. You have to understand just the basics. Again, we've already talked about this before, but the basics of how these vaccines work. Remember, these vaccines have a lipid nanoparticle and they have mRNA that gets taken into the cell. Once it's taken into this cell, what happens? Let's say that this is just some type of host cell. It could be any cell that's taken into your body, this vaccine. Once it's taken in, it releases the mRNA. And you guys remember what we said, the mRNA will then bind with what? It'll kind of hop onto our own ribosomal system. And when it hops onto the ribosomal system, what will it do? It'll translate this mRNA, right? So it'll translate this mRNA. And when it translates it into a portion of what? Well, the main thing that we're trying to code for this mRNA is portions of that S peptide. Okay, so portions of that S peptide. Now, what happens is pieces of that S protein will then do what? Will then get expressed onto our cells. And primarily the one that we said was the big one here is this pathway, the Mhc2 pathway. So what's going to happen is this S peptide, part of it's going to get expressed on this Mhc2 molecule. These Mhc2 molecules are only found on what cells? This is only happening on APCs, antigen presenting cells. What are the antigen presenting cells? This is macrophages, this is B cells, and this is dendritic cells. We understand this, right? Once these MHC2 complexes present that piece of the S peptide that activates your immune system, what type of cells? Do you guys remember? These are your T cells, but we should be specific. These are your naive T helper cells. These naive T helper cells will use their TCR to interact with the antigen and their CD4 molecule to interact with the MHC2 complex. Once this interaction occurs, what happens? It starts releasing cytokines, a bunch of different cytokines. And what these cytokines do is, it causes these naive T cells to start proliferating and making more T cells that are primed and able to recognize that S peptide if they ever come into contact with it again in the future, right? So these are going to be my memory T cells. The other thing that we know what happens here is that these T cells will also again release more cytokines. And these cytokines, what will they do? They'll go and activate another set of cells. And these cells are called B cells. These B cells from these cytokines will get converted into very interesting cells called plasma cells. Plasma cells will then start producing what types of molecules? They will start producing antibodies. And these antibodies are going to be particularly primed that if, for whatever reason, we are infected with the SARS-CoV-2 virus and that S protein gets expressed again, we will have antibodies that can bind to it. So far we have memory T cells that we've built up, and we have plasma cells that we've built up, and we have antibodies that we built up. Now here's one more thing. The B cells, whenever the B cells actually, whenever they're stimulated from these cytokines, they go two pathways. One is to become plasma cells. There's a particular name for these. We call these effector B cells, but we don't really call them that. We call them particularly plasma cells, are the effector B cells. But whenever these cytokines stimulate the B cells, we cause these B cells to differentiate into what's called memory B cells. And these memory B cells present particular types of antibodies on their cell membranes. And guess what these antibodies are primed for? If you ever are exposed to the SARS-CoV-2 and that S protein binds onto that little part there, it's going to activate the B cells. And the B cells will activate your immune system. What are these little B cells called? These are called memory B cells. So, We have memory B cells, we have antibodies, we have memory T cells. This other pathway is the kind of like, controversial pathway, but again, it's a small pathway, not as significant, where a piece of the S peptide can get presented onto Mhc2 molecules. And whenever that's presented, these specific types of cells, called naive cytotoxic T cells, will bring their CD4 protein to interact with the MHC1 and their TCR to interact with that antigen. When this interaction occurs, they become activated and they start releasing cytokines. And these cytokines, what they start doing is they start causing an increased production or replication or proliferation of these naive T cells into kind of primed and ready to roll cytotoxic T cells. So that way, if ever in the future a virus, the SARS-CoV-2 virus infects a tissue cell and exposes a piece of that S peptide on their MHC1 complex, these cytotoxic T cells are ready to go. Now, this same kind of process can occur in somewhat a similar way when someone's infected. They generate a very similar kind of immune response, right? Here's the question. Is this immune response strong enough or able enough to battle off these variants? Well, here's the thing. Let's say, worst case scenario, we take, for example, here you have the virus, right? The virus leads to kind of what's the problematic portion of the virus? The problematic portion, we said, is the S peptide, okay? The S1, S2, and the receptor binding domain. Let's say we take, for example, you take that example of where there's a small change in like the S peptide. Small changes. I still think that these antibodies are going to be able to be effective against this S peptide from any of the variants. There might be a small reduction in efficacy, but for the most part, I still think there's going to be effectiveness here with neutralizing antibodies. Again, we need some more studies, RCTs, to really confirm this. I'm just making my kind of own hypothesis here based on just the basic concepts of immunology. So with that being said, I think one of the big things is what Moderna and Pfizer vaccine does and what your natural immunity does is you generate antibodies that are designed to neutralize the virus. Is that going to be effective? I think for the most part it is. And so that leads to this question. We know that these Moderna, Pfizer vaccines, they had pretty good efficacy. Somewhere around like 95% was around their efficacy. Would there be a drop in efficacy because of these antibodies, these neutralizing antibodies that these vaccines help us to generate? I think yes. I think there could be a minor drop in efficacy, but I don't think it's enough that it's going to drop below like 50% where the vaccine is still not effective. Again, you only need the vaccine to have about a 50% efficacy for it to be FDA approved. I still think that regardless of these variants, I think there will be a drop, but I don't think it's going to be enough that the vaccine still won't be effective. And here's the reason why I would back that up. Even if the neutralizing antibodies weren't good enough, well, guess what else you have? Let's say that this S peptide, the virus, the SARS-CoV-2 variants, infect a cell, express a piece of that S peptide. Guess what else you have that can come and try to eradicate it, besides the neutralizing antibodies? Now you have these cytotoxic T cells, which will come to that cell that's infected with it. Now you have these memory B cells that when activated will help to do what? Will help to produce more antibodies. Well, these memory B cells can become activated and kind of eventually trigger more antibody production. And then you'll have more memory T cells, helper cells, come to the area and also help to fight off this infection. So even if the neutralizing antibodies weren't as effective, you still have memory T cells and memory B cells that should still be good to fight off that SARS-CoV-2 variant, whichever one it may be. Another thing that's really important here is that these Moderna and Pfizer, for the most part I believe it's Moderna, they are looking to make small changes to their vaccines. And so their booster shots will account for maybe these small little mutations within the SARS-CoV-2 virus. So again, the booster shots may be something that also will help to increase that efficacy. Again, trying to make sure that you tailor the S protein within that mRNA a little bit so that it kind of can help to accommodate for these SARS-CoV-2 variants. So again, there's another way that we can have that. So I think that kind of answers the question that at the end of the day, is vaccine still effective? Yes. And I think that they will also their efficacy may drop, but I think it'll go back up with the booster shots. And the second thing is our natural immunity enough, if we've been infected with the original, enough to fight off these variants? I think yes, it is. But again, it kind of comes down to that point of how many antibodies and how much memory cells you generated that may be dependent upon how severe your infection was. But again, that's just hypothesis on my part. The last thing that I want to talk about here is prevention. What is something that we can do? What are the things that we should do to kind of prevent ourselves if by the worst case scenario, again, we're infected with SARS-CoV-2 virus, whether it be the original, whether it be the variants, it should still remain the same. It shouldn't be really any change. And I think the ways that we prevent this primarily comes from, again, making sure that this is obviously common sense, washing your hands, making sure that you're quarantining and isolating if you become infected, okay? And here's the big thing. We already talked about this. We had a video. What else did we say can help to optimize your immune system? Vitamin D. Taking vitamin D is a big one. Another one is vitamin C a little bit. And zinc. And the last thing here is, very important, make sure that you are getting vaccinated. Okay, these are some of the things that I would say, and again, also going off of this, wearing your mask, okay? Wearing your mask, quarantining if you're needing to, washing your hands, making sure that you're kind of still trying to maintain that 6 feet distance, and then optimizing your immune system with these natural kind of vitamins and minerals. I think, and I hope that this answers our questions about the SARS-CoV-2 variants. All right, Ninja Nerds, so I really hope that this made sense. I hope that it helped. I hope that it maybe cleared up any confusion on that SARS-CoV-2 variant topic. Ninja Nerds, we thank you. We love you so much for always supporting us and helping us. Ninja Nerds, as always, until next time.